turbulence, oceanic turbulence, and highly intermittent phytoplankton dynamics. You'll see what I meant by highly intermittent plankton basically distribution, how they distribute. This is my first turbulence, microstructure profiler. This guy free fall, roughly around 50 centimeters per second. I call this TurboMAP. This is the first generation. Tip of this profiler, I got shear probe and this blue light. This is LED fluorescence probe. Okay, this is shear probe, roughly centimeter scale. If you cut this in slice, you get structure like that. This W is basically sinking speed. This probe is axisymmetric airfoil of revolution. Mean speed W, this is a sinking speed, so it'll be roughly 50 centimeters per second. And we have a cross stream velocity, that's a turbulence velocity component. Some of these makes big U that create pressure distribution that generate lift force. That lift force generate electricity because this is a ceramic beam. That's how we detect this little U, because we know W from the pressure probe. So this is one typical signal from microstructure profiler. This is temperature, this is the shear, and this is a temperature gradient. And these are called patchy structure, turbulent patch. And the rate of kinetic energy dissipation, this is something we have to deal with. Below the surface mixed layer, we see these patch. Dissipation is somewhere around 10 to minus 8 to 10 to minus 7. Some of you are not familiar with what this number means, or what's the magnitude of 10 to minus 8. So let me go through very simple minded experiment. I call coffee cup experiment. 10 by 10 centimeter coffee. Suppose you put milk up to the half, pour the coffee, because milk is slightly heavier than coffee. So what you get is two-layer system. I'm going to mix this. So before the mixing, two-layer system is basically stratified condition. This stratification can be explained by buoyancy frequency. Our case, coffee and milk, roughly 3 hertz. Okay, important. And we're gonna mix this coffee and milk at 2 revolutions per second. And I watch it, how long it takes to stop the motion. That was 20 seconds. We go through simple mechanic balance, because kinetic energy, original kinetic energy is lost to two state. Kinetic energy dissipation changing into heat, and potential energy. Potential energy is no more than 20%. So if we assume this is 0, we can calculate or estimate what epsilon should be. This case, epsilon is 10 to the minus 3. I showed you, turbulence patch interior of the ocean is somewhere around 10 to the minus 8 to 10 to the minus 7. Fluctuating velocity of this coffee cup case is about 0.3 meter. N was 3. So make use of these number. We can plot N versus the epsilon from all the observed value from the ocean. Our coffee cup is right there. So interior of the ocean, which is here, nothing like that. So, what, organism living in this condition is not experiencing like coffee cup, okay? I'm going to introduce two important length scale. One is Komolov scale, which is a measure of smallest eddy, the function of epsilon and nu, viscosity, kinetic viscosity. And largest scale is energy containing size. That's a fluctuating velocity and epsilon. That gives us the length scale. And the velocity is URMS. Okay, this is actually observed Komolov scale versus the velocity, well, epsilon versus the velocity. Okay, these are data from below seasonal thermocline. These are rather, coastal area, and that's coffee cup. Again, coffee cup is nothing like you see in interior of the ocean. Okay, phytoplankton require light and nutrient. They need both of them. And diatom and dinoflagellate, single cell, they are much smaller than 1 millimeter scale. However, most biologists use Niskin bottle, which is roughly 1 meter scale, and lower Niskin bottle with cable, you get finite number of data from finite number of points. So people look at distribution of phytoplankton, something like that, and this is well known subsurface maximum. This really representing real distribution. So one of earlier work I did was using this CTD cage that mount fluorescence probe. But this case, light goes here. So, cage goes up and down due to, wave motion. And also this protected, area destroyed, any, undisturbed feature. But my profiler, I have, fluorescence probe looking outside, which doesn't disturb real distribution. And this guy can resolve roughly 2 centimeter scale. So we went to south of Tokyo, this island, and we went to south of this island, dropped, CTD and TurboMAP side by side, and we got this. This is, turbulence data, and red comes from CTD, and blue comes from TurboMAP. So clearly, my LED probe shows more intermittent feature than CTD. But if you average over 1 meter, they are the same, pretty much the same. So if you average over 1 meter, you lose original, intermittent, distribution. Then I came to next question. Is this resolution sufficient? So I developed, another fluorescence probe based on laser diode. This can resolve millimeter scale. In order to, accommodate this, probe, I put both LED and the laser probe on the single profiler. This is called TurboMAP-L, and I have 2 fluorescence probe, LED. This can resolve centimeter scale, laser millimeter scale. Okay, I'm going to stop here. Are you guys okay? Am I going too fast?
No, this is good.
It's good.
Is it good?
Yes, yes, good, good.
Okay. So I have, two fluorescence probe, centimeter scale and millimeter scale. This shows example compared CTD, fluorescence data against LED centimeter scale, and this is millimeter scale. Centimeter scale is quite less intermittent than, laser fluorescence probe, millimeter scale. And also another interesting feature, laser probe detected was, variance is different from layer to layer. LED couldn't resolve that. However, if you average these guys, these two are original data from LED and laser. And this is 2 centimeter scale average. This is 10 centimeter scale. And this is 50 centimeter and 1 meter scale. So again, if you average 1 meter, all the important, highly intermittent features disappear. So 1 meter average agree. Okay, so far I introduced 4 different ways to measure phytoplankton with Niskin bottle and C-point mounted on CTD and LED centimeter scale probe and millimeter scale probe laser. Okay, and I showed you every meter's average, all of them agree. Difference is variance. So you can compute, except Niskin, you can compute mean and variance for each meter scale. Then you can plot mean versus to standard deviation. This is called coefficient of variation. Niskin bottle, there's no standard deviation, so it's only mean. And C-point, small variability, so there's some standard deviation, but much less than LED. But laser is much, much bigger than LED. And coefficient of variation actually exceed this dot is 1. So these are the example of laser. Actually, this is quite extreme case in near the Tokyo Bay, and that's a mean. And the expanded view, we found such a strong signal emanating from mostly these aggregate. I show you why we think that way later. But the signal is extremely strong. But if you average, you get conventional picture. Okay, this is example. We sampled, with TurboMAP from inside of bay toward outside. We're looking at CV, coefficient of variation. Inside of bay, CV was low. Going toward outside, CV goes high, really high. So open water, going toward open water, CV is quite high. In this general feature, so we collected, all kind of, place. We went to all kind of place and we collected, a bunch of data. Even we went to a Santa Barbara channel. We plotted everything we had. This is what we got. Okay, this is a mean. This is a standard deviation. What you're seeing is high salinity in red. These guys are mostly from, bay, estuary, and so forth, near coast. And these guys are coming from actually, open water. These really high, CV, they are associated with Kuroshio. Okay, so we found very, distinct feature in CV, open water type and estuary type. Okay, let's go to this time series again. Having time series, what do you do first? We often, compute spectrum. So I did. Red, this is LED. Green, this is, laser. Less than 1 cycle per meter or bigger than 1 meter scale. This is -5/3, turbulence spectrum, equivalent to turbulence spectrum. But beyond that, red one shows some color, but the green, laser shows pretty much flat white noise. Because red one is LED, we have a resolution, limitation. That's why there's a, coloring feature. Okay, let's look at another feature. What's the distribution of these each signal? What's the PDF? So we generated, this histogram. Clearly, green, LED, pink. Green one is, laser. It's very, very strong signal. So PDF is very, very long tail. So epsilon often appear log normal in the ocean. So we took log of this. LED shows normal distribution after you take, log. But laser, it's not. Still have long tail. So you took one more log. This is called Gamble distribution, or some people call hazardous distribution, because tail is so long. Extreme value can take place very small, probability, but not diminish to 0. It still exists. Okay, let's see if you find the peak of these signal and call these guys as some patch. Let's define these patch based on some threshold value. Okay, one example. This is a threshold value, roughly 10, 2 times, 5 times, mean background. These black points, they are the patch. Let's call patch. If we assume they distribute completely random fashion, how do you expect to encounter these guys? That's a complete random fashion. So it should appear as a, Poisson process. So each event should take place like, exponential distribution. As a matter of fact, green one is observed, CDF. Blue one is, exponential distribution based on the parameter from this, statistic. So indeed, based on this, threshold, event appear completely random fashion. Okay, we lower the, threshold to increase, integrate, include, smaller patch. Then what's going to happen to event? Actually, blue one is, Poisson process, but this is what we observe. actually observed, 90 percentile takes place shorter distance than expected, exponential distribution. What this means is, looks like small one reside around the big one. That's what this suggesting. Okay. From these data, we do see something there, but and we are detecting something. I couldn't see it. So next thing, I want to see it. Then I mounted this camera system using LED as a light source. This distance is roughly 10 centimeter scale. This was originally developed for mammal, experiment to see what they are seeing. I went to this company who makes this, camera system to modify so that I can take some particle, whatever exists, emitted by this, light system, and light emitting from LED, and I'm gonna discretize 2 by 2 centimeter scale. We are getting into the water. Now we see bunch of particles. See, lots of particles. Looks like those are the source of, highly intermittent, fluorescence signal. Now we can go through, image processing, and we can count how many particles we came across. And also we can compute, from shear probe dissipation. We can, average, particle size. So we have an average dissipation rate against average particle size. So we can plot this from bunch of clues. We compose this from based on many, many clues. Looking at dissipation all the way to here is 10 to the minus 5. I want you to pay attention to 10 to the minus 6 here. And this line is Kolmogorov scale based on each dissipation rate. And 10 to the minus 6 here, dissipation 10 to the minus 6. Kolmogorov scale is 1 millimeter. And up to 10 to the minus 6, dissipation enhance average, aggregate size. But beyond this, looks like destruction start happening. So below here, aggregation is more active than disaggregation, but looks like above Kolmogorov scale, this destruction is more active. So we went to next stage based on all the observed, particle size. We constructed, size spectrum. We consistently found two slope. One is not as steep as second slope, and we computed based on average, dissipation rate. Kolmogorov scale right there. So this is consistent. Looks like, destruction is happening below this size, above this size. Below this size, aggregation is, happening. And we consistently saw these two slope for each, campaign. And always this is less steep than that steep slope. Okay, I think I went through quite quickly, but I'm going to summarize my talk. Okay, intermittent phytoplankton signals require at least to millimeter or millimeter scale, large fraction of intermittent phytoplankton signal associated with marine aggregate. Marine aggregates are not dead body. They are actually hotspot, library. Okay, turbulence enhanced the size of marine aggregate up to the Kolmogorov scale. Well, our data shows this evidence, but I'm not sure if we really prove this. But at least, from our data set, this is consistent. And important thing is question the variation we found from, phytoplankton signal is high in open water, such as the Kuroshio. Okay, I didn't introduce what's next. Kuroshio is well known to be oleotrophic, poor nutrient. But from other, numerical work, we found, high CB supports high diversity and high, plankton, ecosystem diversity. High CB may suggest high biodiversity, and since Kuroshio is high CV, Kuroshio is high biodiversity. That's one of our still kind of hypothesis, but I think that's what it's pointing. That's kind of quick summary of my quick talk on my intermittency of phytoplankton. Okay, so we have plenty of time to discuss.
That's great. Thank you very much, Hide. Yes, indeed, we have plenty of time for questions. And please, if you have questions from the audience, just go ahead.
Something I didn't introduce because I thought I don't have time, and also if I have to go through that part, I certainly run out of time, because we found this in intermittent feature. We develop series of numerical model. I show you example, just example. Okay. Okay. We started from this simplest, NP ecosystem model. We found, turbulence study. Basically, we separated, P and N into mean and fluctuating component. You come up with, mean equation and variance and covariance equation. And we went all the way to NP and NPZ and NPZD. And even we went to NPZF. All those, different stage of, model we develop, and consistently pinpointing high CV supports high productivity and high biodiversity. I have a series of papers. These are already published, so I can list all those if you guys are interested in. Okay.
It's great. I have just a quick question, maybe because I don't have background in this. What will be the challenges if you have to develop a probe or like the one you showed, but that goes to the micron scale or smaller scale?
Mm-hmm. Well, the light source has to be small. You know, original the first fluorescence probe I developed is this one. LED probe. There are 6 LED diode, but this light source, of course, compared to these guys, that's smaller. And the important thing is, I'm getting undisturbed, field. That's important. This one, because 6 diode culminated to point, so that makes resolution rather not so small as I wanted. So we found this blue laser. Japanese guy developed at that time blue laser diode. So that was millimeter scale. So we could successfully resolve millimeter scale. Unfortunately, company who made this probe, they stopped manufacturing because nobody bought it, except few people, because those people bought this probe because their you're getting is this sort of highly bunch of signal, strong signal. They thought this is a noise, complete noise. And they didn't even compute, spectrum. Spectrum is this. No, if they compute this is LED, this is the laser, and you see the difference in high wave number. I know this because of resolution. It's a filtering, and this guy is resolving very, very small scale. That's why at least this range of signal is white. That's an interesting thing.
Thank you.
Okay.
I have a question. This is Mimi. What was the spatial scale of your shear measurements that went along with? It's fantastic. You can measure this fine scale in the field in different habitats. That's really neat. I was curious about the shear.
Shear is okay. This is centimeter scale.
Mm.
So we are roughly resolving centimeter scale field. But as far as dissipation goes, because we are not resolving millimeter scale, but we can fit sort of universal spectrum, the shear shear data. So we can extrapolate high wave number part, so we can estimate dissipation.
Yeah, I was just going to mention on not in all these different habitats and over such large depth scales, but we've been using PIV in the field to look at the shear distribution and vorticity distribution, and we find the fine scales that you find with your phytoplankton distribution. So I just thought I'd toss that in, that we see variation on this very fine scale like you're seeing too.
Right.
So yeah, very neat.
Marco?
Yes, so thanks for the talk. I was intrigued by the plots where you look at the coefficient of variation, in particular the differences the different curves that you get for the same hardware, right, in different locations, right?
This one? This one?
This one, yes. So I was wondering whether you have any idea for what is the origin of the different dependence of the standard deviation
Well,
on the
yeah, we know that because these guys, high CV, these guys are from place like Kuroshio. Kuroshio is really, really high CV, which means, strong signal is emanating from very small spot. That's why,
Right. No, I
standard
understand that.
deviation is high, but if you average much lower than these,
Sure,
coastal numbers.
but if I were to go down to actually see what's there at the microscale, what would be the actual differences? I don't know, for example, in population of
This
phytoplankton.
was my not complete proof, but this is my at least consistent picture. These signals are emanating from aggregate, marine aggregate. So within the marine aggregate, phytoplankton, they are library there. They're very, very active library there. They are the one emanating strong signal.
Okay, I see. That's interesting. If I'm allowed, there is a follow-up question on that. Some non-biological material, for example, plastics, they are, for example, plastics, they are fluorescent as well. They're autofluorescent. And so I was wondering whether you might been picking up some signal
I think so.
from these.
Look at here.
In particular,
Can you see?
exactly, in particular in places where you're maybe closer to cities or estuaries or something like that of main
I
rivers.
think so, but actually these two figures are from different kind of camera system. We had CPIX mounted on cable observatory. This is undisturbed real marine aggregate photo, and you see if you look detail, I think all kind of thing in there. But the thing is, live phytoplankton are also here. So plastic, maybe like a microplastic, it could be integrated in marine snow. I don't know.
Okay, thanks.
Am I answering your question?
Yeah, I think so.
These guys appear in different paper, and one another paper we review revising it.
Okay, thanks.
Okay.
Jonas?
Any other question?
Yes.
Yeah, thank you. Thank you for this wonderful talk. I have a question about the meaning of the Poisson fit. Am I interpreting it right that when you say that the extreme peaks obey Poisson statistics, does it just mean that sort of the aggregates are randomly distributed in space?
Okay.
What is the meaning of the Poisson statistics in this context?
If these, let's say these are called event. If event takes place completely random fashion, that means Gaussian fashion. And then Poisson process means event interval should be exponential distribution. That's the Poisson process.
The peak size, or
No,
are we
peak
talking
to
about the
peak.
distribution on the on the depth?
No. Poisson process is looking at interval between each event,
Right.
and what's the distribution of this interval? That's the
Okay.
thing. And
But then
this interval distribute as a exponential distribution.
I see. So I can interpret essentially each aggregate as being randomly positioned in this
Yes.
At this
Then
depth range.
That's what I was showing. If you use high threshold, that means I'm picking up larger aggregate or larger something, and then I showed you
Yes.
observed event interval. This is a CDF form.
Yes.
Pretty much agree with exponential distribution.
And what is as you lower the threshold, this stops
Then,
being exponential, and is it revealing some spatial structure of the distribution?
Okay, I lowered here. Okay, so I'm including smaller aggregate
Yes.
as well. In addition to these big guy. So I have more points.
Yes.
Then green one shows observed CDF, observed interval CDF, cumulative distribution. This is exponential distribution. We can compute mean interval that gives you parameter to compute exponential distribution. So if it's completely random, it should be exponential. So 90 percentile, 90% probability, everything should happen below 42 millimeter if it's completely random. But actually, observed one is shorter than that.
So I'm just trying to visualize this spatially. Would that mean that small events are close to large events?
Yes.
They tend to cluster together?
Yeah.
Okay. Interesting. Thank you.
Looks like, big one maybe, destroyed or, came apart.
Yes.
So small ones still reside nearby big one.
Thank you. Very clear. Thank you.
I think that's the way, this data is pinpointing.
Super.
Okay.
Any more questions? Well, thank you very much, Hide. We really appreciate this. That was very interesting talk and results. And yes,
all these materials I have already published, so I can share with you guys if you like.
Yeah, that's great. We have this. It will be also on YouTube. So if anyone needs the information,
sure.
Yes, we can. We'll have this with the references. That's great.
Okay. Another thing I can do for you guys, if you guys are interested in numerical model, I can list all the published paper, upload to you guys.
Nice. Yes.
Thank you, Hide.
Thank you. This was great.
Thank you.
Thank you very much. Super interesting.
Bye.
Yeah.